Hi, my name is Glennis Irving. It's October 22nd, 2022. I'm doing my presentation on the anatomy and physiology of the retina. So to start, the retina is the sensory organ of the eye, which makes a lot of sense because there's photoreceptors and a lot of cells in this area. A little bit of anatomy of the retina. So the retina was actually developed from the same embryonic tissue as the brain, which is pretty interesting. There are 5 types of neurons in the retinal layers. There's photoreceptors, bipolar cells, ganglion cells, amacrine cells, and horizontal cells. And I'll go into more detail of those cells later. The retina also has glial cells, astrocytes, and Mueller cells, just like in neural tissue. There's a pigment epithelium layer. This layer absorbs light rays that escape the photoreceptors, and it prevents distracting light from reflecting inside the eye. So it prevents like distortion of the visual image. The fovea is also on this layer, or is on the retina. The fovea is free of neurons and blood vessels, and also of rods, so it only contains cones. It forms the center of our visual field, and lens focuses objects on it, and that's how we see objects. So in order for us to understand like how the retina works, we need to understand how the lens works as well, since the lens reflects or focuses light onto the retina. So there's a process called accommodation, which describes how we are able to adjust from seeing far and seeing near. So when an object is more than 20 feet away, our lens is flatter and parallel light rays are created, which will cause the image to be in focus. When an object is closer than 20 feet, light rays are not parallel anymore, and the rays strike the lens at an oblique angle, which changes the distance. This creates the focal point behind the retina, which causes blurriness. So the lens needs to become more rounded in order for that object or image to become more focused. So when the lens is convex, the light rays will converge on retina instead of behind it, and then vision would no longer be blurry or out of focus. The ciliary muscles are what adjust the lens from flat to round shape. And there's the vision defects like hyperopia and myopia. So hyperopia is farsightedness, where the focal point is behind the retina, and corrected lenses would need to be convex. And then for myopia, that's nearsightedness, so the focal point would be in front of the retina and would be corrected with concave lenses. So moving on to phototransduction, this is what takes place when light hits the retina. When light hits the retina, photoreceptors convert the light energy into electrical signals. So this light energy is actually part of the electromagnetic spectrum. So humans can see visible light, which falls between ultraviolet rays and infrared rays. The wavelength of the visible light would be 400 to 750 nanometers, and red, green, and blue are the primary colors that make visible light. So for people with color blindness, they have a difficult time seeing red and blue, or red and green. So then on to photoreceptors. So there's 2 different kinds of photoreceptors. It's rods and cones. Rods function mainly in low light, while cones function in during the daytime. It's like our sharpest vision and the color vision. Rods outnumber cones 20 to 1. So the photoreceptors have different layers, in structure. Their rods and cones, they have a similar layers. So the outer segment would be touching the pigment epithelium. The inner segment contains the nucleus and organelles for ATP and photosynthesis. And then the basal segment has a synaptic terminal, which releases glutamate onto bipolar cells. They also have visual pigments. So these pigments are bound to the disc membrane of the outer segment of the rods and cones. They're transducers, so they convert light energy into a change in membrane potential. Rods have rhodopsin, and cones have 3 different types of pigments, but they're similar to rhodopsin. The pigments in the cones are excited by different wavelengths of light, so that's what allows us to see color. So there's like red, green, and blue pigments. A little fun fact: our brain recognizes color of object by interpreting the combinations of signals from the 3 color cones. So moving forward to transduction for rods. So rhodopsin has 2 molecules, opsin and retinol. Retinol binds to opsin when there's no light. When a photon of light hits the rod, retinol is activated. The retinol no longer binds to opsin and is released from the pigment rhodopsin, which is also called bleaching. Rods have 3 main cation channels. So in the cyclic nucleotide GAA channels, there has sodium and calcium channel that are to enter the rod. There's so for potassium to leak out of the rod, and then calcium regulates exocytosis of the neurotransmitters. So in darkness, rhodopsin is not activated, so during this time, cyclic GMP levels are high, all the channels are open, and glutamate is released to the adjacent bipolar cells. When the light activates rhodopsin, the cGMP levels decrease, and the channels are closed. Inside, the rod becomes hyperpolarized versus when it's in darkness, it's depolarized. The glutamate release decreases, and then the retinol diffuses out of the rod and is transported into the pigment epithelium. So retinol then reverts to inactive from being formed before moving back to the rod to reunite to the opsin and bind back onto the opsin. So signal processing. So convergence means multiple neurons synapse onto a single postsynaptic cell. So 15 to 45 photoreceptors converge on one bipolar neuron, and then multiple bipolar cells innervate one ganglion cell. So this information from hundreds of millions of retinal photoreceptors condensed down to like 1 million axons leaving the eye by the optic nerve. So horizontal cells, they synapse with photoreceptors and bipolar cells. They mediate the lateral inhibition in the retina, and this lateral inhibition enhances the stimulus location and contrast. The amacrine cells modulate information flowing between the bipolar cells and the ganglion cells. So they're kind of like the mediators. Bipolar cells, there's 2 different types of bipolar cells. There's the on cell and the off. signal process of these cells begins when photoreceptors release glutamate onto the bipolar cells. The on cells are activated when glutamate secretion decreases in light, while the off cells are inhibited. The on cells are inhibited by glutamate release in the dark. So on cells have MGLUR6 that hyperpolarizes the cell when receptor binds glutamate in the dark. And then when the MGLUR6 is not activated, the on cells depolarize. Off cells have ionotropic glutamate receptor that opens ion channels and depolarizes off cells in the dark. Ganglion cells are ganglion cells synapse with bipolar cells. They lie on the surface of the retina, so they receive information from areas called visual receptive fields, and our visual acuity is the greatest in these areas. It's how we see the greatest is from this area. Receptive fields are divided into sections, so there's a round center and a donut-shaped surround. A strong contrast is an excitatory or inhibitory response, and then a weak contrast is an intermediate response. Ganglion cells respond most strongly when light is brightest in the center of the field, during on, center, and off surrounds fields. When light is uniform, the retina uses contrast instead of absolute light intensity to recognize objects in the environment. So contrast allows better detection of weak stimuli also. There's different types of ganglion cells. The most common are the M cells and the P cells, which and they're located in the primate retina. The M cells are sensitive to information about movement, and the P cells are sensitive to signals that pertain to form and detail. And that's it.